Okay, this is podcast number 2 for standard 3.2, and in this one we are going to be taking a look at RNA. So RNA is another molecule, another nucleic acid that is like DNA, but it has some significant differences, and its job is to help DNA to make the proteins that our cells need to stay alive. The reason that DNA needs help from RNA is because DNA is too big to leave the nucleus. Okay, DNA is a really large molecule, and it's so big that it has no way of getting out of the nucleus of the cell, so it has a helper, and RNA is that helper to get the message out to the ribosomes where the proteins get made. So without RNA to help it, the message would never leave the nucleus, and then our cells wouldn't be able to make our different proteins. So RNA is also made of nucleotides, just like DNA. We call them ribonucleotides. So they have the 3 main parts. You have ribose. Ribose is the 5-carbon sugar. Okay, so remember in DNA it's deoxyribose, so in RNA it's ribose. There is the phosphate group, and then you also have the nitrogenous base. So the basic structure is the same. The biggest difference here is that instead of deoxyribose, you have ribose. And then with the bases, one of the bases is going to be different kind of base than what you have in DNA. So we'll take a closer look at that. Adenine, uracil, guanine, and cytosine are the bases. So in this case, the RNA has uracil instead of thymine. It's a very similar molecule. It's a pyrimidine, just like thymine, but there are some slight structural differences, and so that is the molecule that is going to be used anytime you have an RNA. So anytime in an RNA molecule you think it should be a T, it will be a U. Okay? So again, RNA is going to use the uracil instead of thymine in its basic structure, but the complementary base pairing is actually going to stay the same. So adenine is going to go with uracil now, and guanine and cytosine are still going to go together. Okay, so let's take a look. So RNA, a couple of differences between RNA and DNA. RNA is a single strand of nucleotides, unlike DNA, which was a double strand. It has the sugar phosphate backbone going all the way down, and then extending off of that backbone from the sugar are your different bases. And an RNA molecule is the length of one gene, and that's between 1,000 and 3,000 nucleotides. So if you take a couple thousand nucleotides all in a row, that makes a gene, and we usually are going to make our RNA one gene at a time. So if you look over here at the RNA molecule, here's your backbone, still the sugar phosphate backbone, just like in DNA, and here you have your nitrogenous bases that are coming off the end. The only difference here is that you do not have a second strand attached to it. There's just the one singular strand. Now, the way that RNA is made in the cell is through a process called transcription. So in transcription, DNA is actually going to get copied over into a complementary strand of RNA. So remember, to complement means it goes the opposite. This is going to happen inside the nucleus of the cell, okay? Because remember, DNA is too big to get out of the nucleus, so this whole process is going to happen in the nucleus. And it happens one gene at a time. So you do not transcribe an entire chromosome or entire giant piece of DNA. You just do one gene at a time, whatever gene is needed at that moment. In order for transcription to occur, we have another enzyme that's going to be involved in this, and this one is RNA polymerase. So just like DNA polymerase was helping to make DNA molecules during replication, RNA polymerase is going to help to make RNA molecules during transcription. All right, so RNA polymerase has a couple of jobs. So first of all, it's going to bind to a DNA molecule, and it's going to unzip it, open it up, and so you have 2 exposed strands of DNA, so much like how the helicase did during replication. One of those 2 strands is going to serve as a template. And that means that the RNA is going to use that strand to be transcribed. The other strand ends up not being used. The RNA polymerase is going to bring the new RNA nucleotides brought in that are going to complement the DNA nucleotides on that template strand. So the strand that is being used for this transcription piece, the RNA nucleotides that complement it are going to be brought in by the RNA polymerase until you make a new RNA strand. So in this case, G and C are still going to complement each other. So if there's a G on the DNA, it's a C in the RNA, C on DNA, G in the RNA. But there's a little difference with the A's and T's. If there's an A on the DNA, it's going to be a U that you end up having on the RNA, because there are no T's in RNA. However, if it's a T that is on the DNA, then you will have an A in the RNA. So a lot of times I kind of make this little table where you've got, here's your DNA on this side, and you have your RNA on this side. Okay, so if you have a G on DNA, it's a C, C, G. Okay, now if it's an A on the DNA, it's going to be a U in RNA. And if it's a T on the DNA, it's going to be an A on the RNA. So something like that can be kind of helpful when we start to transcribe our molecules. Okay, so let's take a look. When the DNA transcribes the RNA, we actually get a couple of different kinds of RNA in this process. Okay. So this transcription is going to happen inside the nucleus of the cell, as we said. So here's a DNA molecule, and here's your RNA polymerase. So it's going to bind to that DNA molecule, and it's going to open it up, unzips it, and here's your new RNA molecule being made. Now notice it's not staying attached to the DNA molecule because it's just making that piece, and its job is going to eventually go out one of these pores and go out into the cytoplasm of the cell. Okay. Here's another picture of that. Here is your original DNA strand, again with the hydrogen bonds holding them together. Your RNA polymerase will bind, unzip those strands, and then it's going to use one of them as the template strand. And here is your new RNA molecule that will be made. And again, it's only going to bind here temporarily, but it's still going to use the ideas of complementary base pairing in order to bring them in. Once the RNA strand is made, it's going to then leave the nucleus. All right, so there are actually 3 types of RNA, and each one of them is going to play a role in protein synthesis. So there is messenger RNA, which we call mRNA for short. There is ribosomal RNA, which is rRNA. And then there is transfer RNA, and that is tRNA. So these 3 different types of RNA are going to have specific jobs to do during the process of making a protein. Okay, we're going to start with messenger RNA. Messenger RNA does just what its name says. It's the one that's going to take the message about what protein to make from the DNA. That's in the nucleus, right? And that's going to get transcribed there. And it's going to take that message out to the ribosomes that are in the cytoplasm so the protein can actually be made. So if DNA is holding on to some information and needs to get it to the ribosome, it's sending that message through the RNA. So you can almost look like the postal service. It's going to send that message through. If you want to send a letter to somebody across the country, you got to send it through the postal service. So here we have mRNA is going to take the message from DNA and send it out to the ribosome, so the ribosome knows what to do. Okay, so this one we are making in our regular process of transcription. So again, the RNA polymerase would bind here. We've got our template strand, also called the coding strand. We make our RNA. That's our messenger RNA, and it's going to leave the nucleus, go out, find a ribosome, and say, hey, let's make this protein together. Now, ribosomal RNA, which is rRNA for short, this is a special type of RNA that actually makes up ribosomes. Ribosomes are made of RNA so that when mRNA comes over, it has something to complement to that it can stick to temporarily. And it makes the whole process of making the protein that much easier. Okay, so this is a special type of RNA that is combined with other proteins into a very specific shape. And your ribosomes actually end up having 2 parts to them. There's a larger part and a smaller part. And where those 2 parts come together, that's where the mRNA can come in that has the message that's going to hold it temporarily. And there's another spot in here for the 3rd type of RNA called tRNA. And that tRNA is going to be involved with bringing in the correct amino acids. So the way that the ribosomes are structured is very specific in order for the process of making the protein to happen. So let's look at tRNA, which stands for transfer RNA. What it's transferring are amino acids. Okay, so they're going to take the correct amino acids over to the ribosome, and those amino acids are going to become part of the new protein that will eventually be made. Transfer RNAs all have this kind of T weird shape to them, and at the bottom they have something called an anticodon, and that's going to complement the codon that's on the mRNA. So when we get into translation, we'll talk more about that. But carrying off the top, they're holding one of the amino acids, and they're actually really specific about what kind of amino acid they can hold, depending on what their anticodon is down here. Okay? So as we get into translation, we'll take a closer look at how these tRNAs actually do their job. Okay, so here's just something that shows you a breakdown of your 3 different types of RNAs. So there's your messenger RNA, right? Also called mRNA, carries instructions from DNA to the ribosomes. Okay, ribosomal RNA combines with proteins to make up your ribosomes. Okay, transfer RNA is going to bring the amino acids to the ribosomes so that they can become the correct proteins. All right, the very last thing we're going to do in this podcast is we're just going to do a quick comparison of DNA and RNA. This is a really common type of thing to see on test questions and stuff. So remember, DNA is 2-stranded. We call it doubled, a double helix. It's got deoxyribose as its sugar, and it has the base thymine. Whereas when we look at RNA, instead of being 2-stranded, RNA has a single strand. It's also much smaller. Instead of deoxyribose, the sugar RNA is ribose. And instead of thymine, it has a base called uracil. So over here you can see a picture of the DNA molecule with its double helix, and all of the bases are held together by their hydrogen bonds. And you've got A, T, G, and C. Down here is not the best picture, but it's a little picture of an RNA molecule. Single strand, much smaller, and your bases are just extending off to one side. Okay, so one more picture to look at with this. Here again is your DNA, the big giant double helix, and this would continue going on and on forever, whereas RNA, 1 strand. Okay, it can take the shape if it's messenger RNA, but it's only, remember, going to be the length of 1 gene, so it doesn't just keep going on for millions and millions of base pairs. It's going to stop. And then here is our big difference between the bases. You've got uracil here, and you have thymine over here. Okay, and then you can remember the third one by their names. Deoxyribo is deoxyribose. Ribo is the ribose.